Thank you very much. It's a pleasure to be here with you today, and thank you for inviting me and for supporting our work. So I'm going to give you a sort of overview of the models that we use in the lab, why they're important, and sort of an overview of why I came to apply these models to the study of brain cancer. And then I'm going to show you a little bit of work on the specific projects that the Brain Tumor Foundation of Canada is supporting. So here we go. So this is the problem that we're all talking about this weekend. And this is a CT scan of the head of a patient who has a very aggressive tumor, a glioblastoma. And you can see how this glioblastoma doesn't even respect the normal anatomy or symmetry of the brain. It's in the right frontal lobe, opposite on CT, and it's crossing over the midline, infiltrating the corpus callosum, the white matter that crosses from one side of the brain to the other, and it's infiltrating the other side. And if we could actually study this patient's brain with a marker for a cancer cell, say for example a reporter labeled green, then you would actually see the entire brain, both hemispheres, would light up with green cells. And we know this from mouse studies. So it's quite alarming to know that the problem, even with the best surgical excision, if we were able to remove that mass that we see that's visible to the naked eye and visible under the microscope to the surgeon, it's still not going to cure this problem. This tumor is invasive, it's insidious, and it spreads through the brain. So how do we tackle this kind of problem? So I'm going to tell you the story for why I became interested in even studying brain tumors. So during the second year of my residency at the Hospital for Sick Children at University of Toronto, I met two young boys, and they were both named Christopher. And they were both 5 years old, and they were both my inpatients on the ward that I was taking care of. And both of them had a childhood brain tumor. And they had something that occurred in their cerebellar region. They both presented, you can see the mass here that's in the back of the brain, the white lump that shouldn't be there. And both of these boys had a very similar tumor, a medulloblastoma, that blocked the fluid from circulating around their brain, caused the fluid to blow up on the top part of the brain, caused symptoms of vomiting, and the location of the tumor also produced some gait instability, clumsiness. And so the children came into emergency, underwent MRI scans, and the tumors were found. So both of these boys underwent the gold standard treatment, which was surgical resection. The pathologist examined both of the tumors and found that they had the same histopathology. They were both a classic subtype of medulloblastoma. And both of them underwent the exact same protocols of chemotherapy and radiation. So the same protocols, the same doses, the same drugs, and the same amount of radiation. So we took the best possible care of both of them. Everything in our clinical arsenal we had to give to the children, we gave to both boys. And what really struck me at the time is, in caring for these boys and becoming part of their extended family, it was alarming for me that one Christopher died at 15 months post-diagnosis. He succumbed to relapse of the tumor, and there was nothing further we could do to treat him. The other Christopher was alive at the time I finished my residency, and he's probably in university now. So for me, this really struck me as a huge, huge paradox, and something that no matter how much I studied neurosurgery, and no matter how much I studied any type of clinical medicine, the answer wouldn't lie there as to why one Christopher died and why one Christopher survived. And at that point, I realized that if I really wanted to understand the difference between these two tumors, which appeared to be the same by all of our classification systems, I would have to study molecular biology, and I would have to do more work and more research to truly understand what was driving these tumors. This is the question I'm constantly asked by parents, by children themselves, and by other people who are studying brain cancer. Why? And you've heard that question come up today. Why did my uncle get it? Why did I get it? You know? And at this point in time, we just don't have a good answer. And certainly we can look to the Greek philosopher Heraclitus, who said, "One cannot step in the same river twice." And part of the problem in not being able to answer that question well for each patient who asks me that is because of the problem I'd mentioned in the earlier session, and that's heterogeneity. So the problem is even tumors that look like they're the exact same tumor that occur in patients are actually vastly different beasts. And they're so different that that in fact they're different in ways that we can't recognize. And in fact, the problem with cancer is we recognize now more and more that cancer is not a static disease, it's a dynamic disease, and it's evolving within the patient as we speak. As we're sampling the tumor is changing within the person. And so we're never able to capture or pinpoint it. It's like the Heisenberg uncertainty principle. And Dr. Gunning made some jokes about biologists, but I think you should be lucky there isn't a physicist in the room, because a physicist actually trumps a chemist and a biologist. So the two things I remember from physics I've been able to apply to my research. So one of them is the Heisenberg uncertainty principle. And that principle suggests that a molecule or a cell that's in motion, that's constantly in motion, can never be pinpointed in the same point in space or time, because it's constantly in motion, and that means that you can't capture it. So basically, the way we're studying cancer right now is we're taking snapshots of the disease at certain points in time, but we can't really formulate what's going on inside a patient from one snapshot. So how do we capture this dynamic, evolving nature of cancer when we're trying to target it with a drug? We talk a lot about drug targeting, but first we have to know what exactly is it that we're targeting before we move on to find targeted therapies. So I'm going to shift gears a bit and tell you about the properties of a stem cell. Stem cells are also dynamic cells that may actually not be the same cells that we're tracking throughout time, but we think more and more now that the stem cell is actually more of a state than a cell. And by that I mean that certain cells can be driven by their microenvironment or by the factors that surround them into a stem-like state as almost a protective manner to further or allow their survival. Stem cells are very powerful cells. They possess these abilities that almost no other cells in the body can possess. So there are two types of stem cells. One are the embryonic stem cells, which are pluripotent, meaning that they're powerful enough to make an entire embryo or organism. Those aren't the ones we'll be talking about today. We're going to be talking about tissue-specific or somatic stem cells, which are stem cells that are capable of giving rise to an entire organ or tissue. So neural stem cells are capable of giving rise to all of the different lineages that make up the brain. So if you have one neural stem cell, that can generate the billions and trillions of cells that actually make up the brain, every single distinct cell. And that property is known as multilineage differentiation. So the type of division that stem cells undergo is also very special. It's not just dividing into two. So a lot of cells undergo mitosis. You guys all remember grade 9 biology at school? So mitosis is simply the partitioning of DNA and dividing of a cell into two separate cells. But self-renewal is a different type of division that only stem cells can undertake. And in that division, the stem cell is able to make an exact phenocopy of itself and is also able to give rise to a second daughter cell that, depending on the conditions of the microenvironment, that second daughter cell can be another stem cell, it can be a progenitor cell, which is a slightly more, more differentiated cell than a stem cell, or it can be a completely mature cell that makes up the brain, like an astrocyte, which is a supportive cell in the brain, or a neuron, which is of course the cell that fires signals and allows us to think. So the fact is that all of these cells can arise from a single neural stem cell, and this property is exclusive to stem cells. Stem cells can also proliferate, but further to that, they have a property called quiescence, which allows them to pull back from the cell cycle and hide out in a phase of the cell cycle where they're usually not actively proliferating or detected. This is important. If we skip to later in the talk, I'm going to tell you that chemotherapy and radiation therapy, which are our mainstays, only target rapidly proliferating cells. They're not able to capture cells that are hiding out in a non-proliferative state. So possibly more important than proliferation is the stem cell's ability to hide out in a quiescent state. The last property is longevity. So why is that important? If you think about other cells in our body turn over quite rapidly. Blood cells only live for 90 days. Other cells are quite transient in their lifespan. But a stem cell can actually exist for the lifetime of an organism. So all the neural stem cells we're born with, thankfully, actually aren't the only ones that are there. They do regenerate and form more stem cells later in our lives, but the stem cells we're born with are capable of persisting until we're elderly. And so if you think about that in the context of cancer, that actually makes those cells sitting ducks for accumulation of mutations that may drive cancer, because they're cells that are around for so long, they're around to accumulate mutations. So you can recognize that on the flip side of all these very powerful and good properties of a stem cell, what I always tell my students is to think, imagine what happens if this process, which is so tightly regulated during development, goes wrong. So whenever I teach developmental biology or biochemistry or anything, I always tell the students, perturb the system. What happens if this goes wrong? And so in this case, you can see that the normal properties of a stem cell, if you flip them all, then what you have is uncontrolled self-renewal, uncontrolled growth, and a blockage of differentiation causing cells to only go down one pathway and generate too many of, say, astrocytes in an astrocytoma. And a cell that's around that becomes immortal and is bearing mutations is a very dangerous cell. So the flip side of all these good properties is actually cancer. So the place that we learn the most about stem cells is actually from the blood system. And here we have a very great legacy. So up on the top there you see James Till and Ernest McCulloch. They are two Canadian scientists who I really hope will win the Nobel Prize soon. They've already been nominated for the Gardner. So they were both working at Princess Margaret Hospital in Toronto in the late 1950s. Jim Till is a radiation physicist. Ernest McCulloch was a hematologist, oncologist. And so those are not two people that would ever have talked to each other in 1950s Toronto. But they happened to be collaborating because Dr. McCulloch wanted to know the effects of radiation on his patients, and so they radiated a bunch of mice. And they happened to notice that there were little bumps on the spleen of the mice. And Dr. Till made that observation when they were sacrificing the animals. This just shows you how scientific discovery is often about serendipity. You're looking for something, and sometimes the side observation is actually what's going to lead you to something great. So it turned out that those little bumps on the spleen were actually colony-forming units, and at that time, when you took out the cells that formed those little bumps, that's when Dr. Till and McCulloch discovered stem cells. So that was the first discovery of hematopoietic stem cells. And so making that discovery back in the late 1950s, they had plenty of time to flesh out the hierarchy of stem cells. So here on the right side, you can see that the entire stem cell hierarchy exists from that cell when it divides originally at the beginning, giving rise to all of the daughter cells that eventually can differentiate into cells to make up all the different lineages of the blood. So there on the far right, you see platelets and white blood cells and red blood cells. All of those cells arose from that one stem cell, the red one at the top of the hierarchy. So we learned all of this from these researchers who I'll mention to you later. All of them are Canadian, but they did all of this great work in identifying markers that specifically or uniquely identified each of these different lineages, allowing us to really see how the blood is organized. And you can see the nice thing about studying stem cells is that it's a way to organize chaos. You could have 1 million cells, and really the stem cell hierarchy provides you a way of being able to organize the cells and understand their functionality. So you can start to understand that tissues are organized as a functional hierarchy that originates from stem cells. So you have the stem cell at the top of the hierarchy, the blue cell on the left, that has the rare and unique property of self-renewal, but it divides very slowly and can often remain quiescent. Then it gives rise to progenitor cells, which divide very rapidly, but they have a very short lifespan. They have a rapid burst of proliferation, and then they tend to sort of peter out. And the progenitors will give rise to the differentiated cells that don't proliferate, but form up all of the functional cells that are needed in the brain. And in fact, the human brain does have stem cells, and the reason I'm even mentioning this is that there was a dogma, again back in the 1950s and 60s, that all of the neural stem cells you had were the ones you were born with, and the stem cells would never be able to regenerate or make new copies of themselves. So there were scientists in the 1960s that discovered stem cells existed in the canary forebrain, and those stem cells were capable of generating cells that retain the memory for the birds to acquire new songs. And in that study, it sort of emerged that, oh, actually stem cells are formed during the lifetime of the organism. And the stem cells reside right here. Let's see, oops, I'm not getting the laser pointer working here. The stem cells reside right here. And that region is called the subventricular zone of the brain. And in fact, it's very interesting to note that the subventricular zone, which is called the subependymal zone in the embryo, in that region, we find that if we give mice chemicals, a chemical, for example, a toxic chemical called ENU, then we find that the tumors that arise in the mice all arise in the region of the subventricular zone. So it seems to be there's something about that zone where the stem cells live that's permissive to transformation events or events that can cause cancer. So this work that I'm showing you here is the very first model that was used to actually study these neural stem cells, again developed by a Canadian called Sam Weiss, who's currently at the University of in Calgary in Alberta. And he actually was the first person to be able to develop a model system whereby he harvested the neural stem cells from the subventricular zone, discovered a media that didn't have any serum in it, which is an unusual way to grow cells. And what he found is that 99% of the cells died out in the culture media when there was no serum in it. But the 1% that thrived were the stem cells. And the stem cells form these beautiful clonal floating colonies called neurospheres. And the beautiful things about these neurospheres is if you go back to this hierarchy here, oh, I'm really not going the right direction at all. If you go back to this hierarchy here, imagine that these cells instead of blood are now neural, and you collapse that whole hierarchy and put it all into a ball, what you would have is a neurosphere. You would have a collection of not only the stem cell, but 10,000 other cells that are all of the other daughter cells of that stem cell. So that's a clonal colony, and that provides us with a really, really useful way to study things. Because if I have a tumor that I dissociate and plate into media, and then I see 60 spheres, I know that there's 60 stem cells there. And in fact, it's a clonal assay, a way that I can actually estimate the frequency of the stem cells within a population of cells overall. So this assay was a big breakthrough in permitting people to use stem cell models to study normal development and later cancer. So you get the idea. Here's our rudimentary neural hierarchy. We're nowhere near as sophisticated as the blood people. They have 50 or 60 years on us. But you can see here that we think that there's a neural stem cell at the top of the hierarchy that expresses the markers nestin and CD133. And depending on what growth factors that cell sees in its microenvironment, it can go down and differentiate and give rise to either the lineage of neurons, astrocytes, or oligodendrocytes, the three main types of cells that make up the brain. And in fact, if you take that one stem cell and you put it into this clonal colony and you pick that one sphere, you can differentiate that sphere and it'll give rise to all of the different lineages that make up the brain. So you're starting to understand now that normal neural stem cells are very tightly regulated, and in terms of the development, in terms of both time and location, the cells receive very important signals from the other cells around them. That's one thing. But also, the timing of development is perfectly regulated such that neural stem cells can give rise to a perfect symmetrical brain. But again, if we flip that and think, what happens if you perturb the system? What happens if that neural stem cell acquires a mutation? What happens if that neural stem cell loses the regulation of some of its capacities? It's possible then that that neural stem cell could be transformed into a cancer stem cell. So cancer stem cells, we think, by virtue of mutations acquired in normal stem cells, are dysregulated, and they have a really amped up ability to self-renew and proliferate, and their differentiation is blocked down one lineage. It's like getting a switch stuck in one position, such that instead of generating 3 lineages, you only get 1. And then in this manner, you can generate a brain tumor. This idea is not new, the concept that cancer could arise from a stem cell. And in fact, there's a paper from 1944 that made that observation I told you about, that tumors in the brain seem to all cluster in that area of the subventricular zone. So as far back as the 1940s, people were already making these observations. The more you do science, the more you learn that none of your ideas are new. They're actually all old ideas that are reinvented. So here's the stem cell hierarchy, and I'm telling you that this is the compartment from which cancer arises. So I'm telling you that this is the compartment rather than the others, and we went over the properties. Why? Why the stem cell compartment? Well, it really lies in the major fact of 2 of the properties of a stem cell. The first I already alluded to. The fact that the stem cell is around for the lifetime of the organism allows it to accumulate those cancer-causing mutations. Secondly, the stem cell already has self-renewal machinery. It doesn't have to acquire it de novo. And the body is actually a very efficient system. And if cells already possess a capacity, it's better that that self-renewal machinery be hijacked and taken over by cancer, rather than a cell having to expend energy to generate a whole new set of self-renewal machinery. So that machinery is already there, ready to be hijacked. So we talked a little bit about heterogeneity before, and this is an observation that I made on my neuropathology rotation just after I met the Christophers, just before I started my PhD in the lab. And so what I noticed when I had to stare down a microscope for 10 hours a day and didn't get to talk to anybody and just had to sit and look at cells all day, I noticed that medulloblastoma looks very uniform, just like there was no distinction between the tumors and the 2 Christophers. When you look at those cells, they all look the same. It looks like they're not different at all. But when you stain the cells for markers of different lineages, like a neural lineage on the left or an astrocytic lineage on the right, you notice that there actually are many different lineages expressed within this tumor. So those cells are not identical. They all have different capacities and abilities. And this heterogeneity is essential to understanding the cancer stem cell hypothesis. So in the model on the left, you can see that tumors are heterogeneous. They're made up of different types of cells, but every cell is capable of acquiring mutations that could give rise to a tumor. Whereas in the cancer stem cell model on the right, you see that tumors are heterogeneous, but there's only a rare fraction of cells, the yellow cells, that are capable, that have the machinery, that acquire the mutations to be able to drive the cancer. And in fact, the other proportion of cells, up to 80 or 90%, are there to support the cancer stem cell. They're not necessarily generating the tumor themselves. Perhaps they're just forming a niche to allow the cancer stem cell to thrive. So this is the underpinnings of my PhD thesis. When I went into the lab now, I was ready to explore these questions to try to understand what made one boy's medulloblastoma different from another. And so these are the main principles of my thesis. I tried to understand if brain tumors could possibly originate from normal brain stem cells. I wanted to understand which cell within this obviously heterogeneous tumor was actually driving and maintaining the tumor. And I wanted to know if brain tumors were also organized as a hierarchical stem cell system. And I did my PhD with Peter Dirks at the Hospital for Sick Children, and he's featured there in the corner. So I was lucky enough to have this scientist sit on my PhD committee, because it seems like there's a lot of serendipity in science, and John Dick was interested in our project and available at the time. John Dick is also a very, very renowned researcher and a Canada Research Chair in the study of human leukemia, and he actually discovered in 1994 that there are rare leukemic stem cells that exclusively drive leukemia. So this was a perfect person to sit on my committee and advise me about whether the same model may have applied to brain cancer. So this is the first work that we published where we actually developed a model system whereby we borrowed from all of the other scientists that I showed you. Very often taking ideas from outside of your box and bringing them into your box is a good way to rejuvenate the current model systems. And so in this work, we were able to take brain tumors directly from patients. And what I would do is there's a bridge between the Hospital for Sick Children and the Arthur and Sonia Labatt Brain Tumor Research Center. I would run to the OR on the second floor. I would collect tumors that the surgeons resected. I would run across the bridge to the lab. I was always running. Didn't want to waste any cells or let any cells die. Ran across the bridge, and I would go straight into the lab, and I would dissociate these tumors, enzymatically dissociate them, and then get them into some serum-free media. And disappointingly, 90% of the cells would always die, and I would have to wait patiently and nurture and baby these cells so that I could get the 10% or less of the stem cells to grow out. And when they did, that first time, I remember the first time I saw a dish filled with these beautiful spheres, and I remember bursting out of the door of the tissue culture hood, the room where the hoods were, and running down the hallway yelling "Peter" at the top of my lungs and trying to find him, because at that moment, I think I had realized that there were stem cells in brain tumors. And the proof was seeing these beautiful clonal colonies floating there. And in fact, they weren't just in one brain tumor, they were in 12 different pathological subtypes of brain tumors. And the interesting thing we began to notice is that the more frequent the colonies, the more the percentage of stem cells, the more malignant the tumors. So you see the very benign tumors have a rare fraction of stem cells. The very aggressive tumors have a high fraction of stem cells. And so what we needed to do at this point is find a way to prospectively identify these stem cells so that I could actually isolate them and study them by themselves without all those other cluster of other cells around them. And so again, we turned to the hematopoietic literature where John Dick was using this cell surface marker to isolate his human leukemic stem cells. CD133 was a cell surface receptor which is clearly involved in some signaling pathway, and it's expressed and highly enriched for in the primary human neural stem cells. And so we reasoned that since CD133 enriches for a stem cell population in normal human fetal brain, perhaps it also enriched for stem cells within brain tumors. And indeed, we found that the CD133 expression index almost exactly mirrored that clonal or self-renewal index that I showed you on the previous slides. The more aggressive the tumor, the higher the level of CD133. And in fact, this was another sort of aha moment for me when I actually managed to sort the cells, which, remember, we're using hematopoietic technology. And if you think about it, sorting cells means that you put cells through this fantastic machine called a flow cytometer, which is a combination of lasers and fluidics. And what it is that you label the cells with a certain marker that has a fluorophore or a glowing signal attached to it. And then you run the cells through this fluidic system where the cells are sent one cell at a time. And lasers are focused on the cells to identify the cells that fluoresce or give off the signal versus the ones that don't. And then the fluidic system captures those cells in different compartments and drops them into separate tubes. So you have one tube that has your positive cells and one tube that has your negative cells. So it's a wonderful piece of machinery, but it's meant for blood because blood flows. Blood is used to being liquid, but brain tumors are not used to being liquid. So you can only imagine the amount of technical troubleshooting we had to do to get the brain cells to flow in liquid and to actually be able to be studied in this way. So after all that troubleshooting, this is the result that we got. And you can see that the CD133 positive cells formed these beautiful clonal colonies that are all originating from stem cells, and the negative cells just plated down on the bottom of the dish and differentiated into astrocytes or neurons. So the fascinating thing we realized at that point is that CD133 does enrich or purify for stem cells. It was a marker we could use to pull out the stem cells. This was very exciting for us. One of my smarter colleagues, another neurosurgery resident called Todd Mainprize, said to me one day, how do you know that you're not just capturing normal neural stem cells that migrated into the tumor? Because we know normal neural stem cells like to migrate to areas of injury. So how do I know that that whole time I thought I was studying tumor stem cells, but I was just studying normal ones? And I said, wow, that's a good point, Todd. I better do something to prove that it really is cancer cells. So we did this assay called spectral karyotyping, and I can assure you that those cells were not normal cells because they have all these chromosomal rearrangements and mutations and problems that we only see in cancer. So this assay made sure that we were really studying stem cells that came from the brain tumor itself. And in fact, the interesting thing is that both the positive and the negative cells had the same chromosomal aberrations, indicating that they were likely arising from each other in a type of clonal hierarchy. So the CD133 positive cells were giving rise to more positive cells, and perhaps also their second daughter cell were negatives. So the gold standard test for anything is to be able to put it into a mouse. And to really understand if these form tumors, I'd have to do more than just look at cells in a dish. So we got immunocompromised mice, and there's a beautiful mouse imaging center at the Hospital for Sick Children where they do MRIs on mice who are anesthetized and taken care of after they've been implanted with cells from human brain tumors. So we would isolate the stem cells, sort them into the CD133 positive and negative cells, and then transplant them into the right frontal lobe of these immunocompromised mice. And we would take them over to the MR to image them. They were imaged in a rather exciting way. We would take 12 mice, all anesthetized, and they would all get put into a little sort of slide that looked like they were about to go down a luge course, and then they would stack the mice into a pyramid of little luge slides, and then we'd image 12 mice at once. And so we did that time and time and time again to document what was happening here. And what we found is, here you can see an MRI, and look at the tumor, the white mass that's formed there. It's formed in the subventricular zone, which is not where I injected the cells. So clearly that's where they like to reside, where the other neural stem cells live. And you see that this is an injection of pure CD133 positive cells. And we went down as low as 100 cells, and we were able to see tumors form. Whereas when we injected up to 100,000 CD133 negative cells, we never saw tumors in any of the brains at all. And when we examined the histology of these tumors, we saw that they were actually identical, really identical to the patient tumors. And here you can see the xenograft from the mouse and the patient tumor. They have all the classical features that pathologists would use to diagnose medulloblastoma. And the really interesting thing here is that cancer cell lines, we study in cancer research, 90% of us use cancer cell lines, which are cells that are passaged over long term and usually harvested from a patient and cultured until they're immortalized, and they've been in culture for decades. And those cells, they don't look like patient tumors anymore. They've acquired all kinds of artifacts in culture. And in fact, they don't look anything like the tumors I take out of patients. So we were really encouraged by this, that the phenotype of our tumors exactly resembled that of the patients. It made us realize that this was a good model to use to study brain cancer. So basically what I've shown here is that we were able to isolate this stem cell populations from brain tumors using the cell surface marker CD133. The tumor-initiating CD133 positive cell has a very, very potent ability to form tumors in vivo, and also can form tumors from as few as 100 cells. And those tumors generated only by the positive cells looked exactly like patient tumors. What are the clinical implications of this? Well, our traditional bulk analysis of tumors is perhaps not the way to go about it. If 90% of the cells within a tumor, 70 to 90% of cells within a tumor, are not the stem cells and not the ones generating the tumor, is there any point in profiling their mutations and looking at how they're functioning? Or should we perhaps redirect our methods of diagnosis and therapy against the cells that are actually generating the tumor? The frightening thing about this is that they may be different populations from patient to patient. We should never make assumptions that the same cells, that the same mutations or the same cell populations are driving cancer in any given patient. So this kind of idea back in 2003 when we did this work took hold, and other models, other labs corroborated the work. And in fact, this work had very new therapeutic implications. And here's some work that came out of Jeremy Rich's lab, where he showed that the glioma stem cells isolated with CD133 were actually radioresistant. And many other studies were done to show that they were also chemoresistant. So what we're realizing now is it's these CD133 positive cells that are surviving through chemotherapy and radiation therapy, perhaps using enhanced mechanisms of repairing DNA that stem cells have, perhaps hiding out in a quiescent phase and avoiding the therapy targeted against them. In any case, there's some mechanism that these stem cells have to evade therapy, and they come back to drive the relapse. So the problem is that these cancer stem cells are treated with radiation and chemo, and they see all those treatments, but for some reason they have mechanisms that allow them to evade this therapy, and then the tumor, which originally after surgery looks like it's shrinking and, oh look, we're getting a good response from chemo and radiation, but then with, for example, with glioblastoma, we know that invariably, inevitably, 5 months after the treatment is finished, the tumor starts to explode again. Why is that? Because we haven't eradicated the cancer stem cells. So at my institute is designed with one mandate. Our mandate is to study, using human stem cell model systems, mechanisms that regulate normal development that might be dysregulated in cancer. So it's a wonderful place where I have people who, in parallel to me, study normal neural stem cells, and I study brain cancer stem cells, and the two of us talk about what has gone wrong and what do we perturb in the system to cause brain cancer. So we very much apply developmental neurobiology models to the study of brain cancer. And this is actually my lab, and you can see the 3 programs that were described in the introduction. I really want to emphasize here that our medulloblastoma program, our 2 students who have done all kinds of beautiful and promising research, are actually the 2 students who were both funded by BTFC studentships. So pretty much you guys realize that I'm not in the lab doing the work myself with my hands on medulloblastoma, but these 2 guys are. And so if there's any great discoveries that are made, I think we'll be thanking you first. So here's an example of the spheres that we culture from all the different tumors that we get from patients. We get over 100 tumors a year at our lab. The McMaster Children's Hospital services a catchment area of 2.4 million people. I get all the tumors from my adult neurosurgeons and colleagues as well. So we get over 100 tumors a year. So to this point in time, we've accumulated, I've been running for 7 years, over 700 tumors. And so you can see here tumor spheres from at the very top. That's an anaplastic medulloblastoma. The second one is a metastatic melanoma. We all know how aggressive that cancer is. I want you to note the number of spheres and the frequency of stem cells in that tumor. And at the bottom here, you see tumor spheres from a glioblastoma. So we have all these cells constantly growing directly from patient brain tumors in our lab all the time, and we're using very different model systems I'm going to show you now to try to understand the biology of what drives these tumors. So here's one of my patients who comes in with a 2-week history of vomiting and gait problems, and he goes through the same diagnostic as the Christophers now, and he's diagnosed with medulloblastoma. So this is the most common malignant brain tumor in children, and I think everyone in this room has seen a slide like this before because you all have heard so much about pediatric brain tumors. But the problem with medulloblastoma is in the 3rd bullet point. The problem is that we're able to treat the tumor locally with surgery and radiation and chemo. But when children recur with medulloblastoma, they recur most often in a pattern where they drop cells down the spinal axis through the cerebrospinal fluid, and they get spinal metastases. And when when that happens, we have almost no therapeutics to offer them, and very often we're making them palliative. And so the real question here is, how do we find the cells that are driving that metastasis, that are driving that spread of the cells down the spinal axis, and how do we block it? So again, here are the model systems that we use in the lab. We get the brain tumors from the patients, we dissociate the tumors and we culture them in the serum-free media to grow them as tumor spheres. We're then able to profile and study the tumor spheres, and we're able to purify them, sort them through our own flow cytometer that we have in my lab, and we're able to inject them into non-skid mice where they form tumors that we're then able to profile and study. And here's an example of a method of what we call genetic engineering, and it's a way that we're able to modify the cells according to the biology we think that is driving cancer. So this is called a lentivector, and it's a type of a virus that we can use that won't make anybody sick. It's replication incompetent, so in other words, it's not going to drive itself to give anybody an illness. But what we're able to do with these vectors is we can knock genes into this construct that we want to express in a brain tumor, or we can design ways to knock down genes that we think are important in driving brain tumorigenesis. So we package up these vectors, we transduce them into the brain tumor stem cells, and then we're able to read out all of the different aspects of the brain tumor stem cells, their genetic gene expression, their functionality, their self-renewal index in the assays we do in the lab, and we're able to try to understand how does that gene regulate the function of the tumor. So this is a very important way of using genetic engineering approaches to try to understand what a gene is actually doing to the function of the cells and to the genetic profile of the cells. And so here's an example. We've published on the master regulatory stem cell gene BMI1. It's a very important gene that regulates normal neural stem cell function. So we said, what happens if we knock down BMI1? How will it affect the stem cell populations in brain tumors? And in fact, we're able to show that when BMI1 was knocked down, the brain tumors became much less invasive, much smaller, and very, very circumscribed. So this tells us that BMI1 is an important regulator of brain tumor stem cells. And in fact, this work led us to many other genes that we think are also important in regulating brain tumors. But the essential role of my lab is to try to understand how these master stem cell genes that regulate normal neural stem cells, if they're really that powerful, then are they doing something to cause brain tumors to form? And in fact, we have models that involve not only studying the CD133 positive cells, but also the CD133 negative cells. So 90% of cells in the tumor, they must be doing something. It turns out that they're actually supporting the CD133 positive cells by secreting important signals and growth factors to them in order to allow the CD133 positive cells to survive. So the interesting thing about a brain tumor is that if we can start to dissect out the sort of chaos of a brain tumor, we can start to understand that some cells are forming supportive niches to allow other cells to drive the tumor to grow. And if we can understand the downstream signaling mechanisms of how all of this happens, we can design drugs to block this process. So the problem I mentioned to you before is that when we biopsy a brain tumor from a patient, we take out the brain tumor and we study it, but then in the meantime the patient goes for chemo and radiation and the tumor's changing in the patient as we speak, and the piece of tumor we have is almost historical. It doesn't any longer represent the tumor that's growing in the patient. So what's the point of studying tissue that no longer represents or captures the tumor that's evolving in the patient? We wanted to address this important problem of how to develop a model system that reflects the dynamic nature of cancer. And so we developed this new model system in my lab, which is, I've submitted this for funding to CIHR, and we've been working on it for a couple of years now. So this project is called Clonal Evolution of Medulloblastoma Stem Cells in Response to Therapy. So I took the COG protocols that we use to treat my own patients for medulloblastoma, and we adapted them for immunocompromised mice. So this sounds like a kind of stupid thing to do because the mice are already immunocompromised, and now I'm going to treat them with chemo and radiation, and they hardly have immune systems to begin with, so how are they not all going to die of infections? We had to troubleshoot all of that for years. And finally, we were able to adapt the doses and find ways using our wonderful veterinary colleagues to actually keep the mice going through their treatment. So what we do is we take the human brain tumors, we dissociate them, we take the stem cells and sort them into different brain tumor stem cell fractions and implant them into the brains of non-skid mice. Then we treat the mice with the same protocols of chemo and radiation that the children get. And now what we're able to do with mice is we can sacrifice them at different time points, and we can study the tumor at different stages of treatment, and we can profile all of the stem cell populations at every time point. And the goal of this model is to try to understand which stem cell populations are still there after the first dose of chemotherapy, which stem cell populations are there after we give radiation, and how are they changing over time. So this is a way to actually try to track all of the different clones of stem cells as they evolve through therapy. And so using this model system, we're able to see that, and this data is a bit frightening actually, because what we see is that after we treat these tumors with radiation, we see a big enrichment in the various stem cell populations. So this really validates the fact that these stem cell populations are actually escaping therapy. And what we're seeing is, rather than the chemo and the radiation killing off those cells, those populations are actually expanding and growing and thriving because they're somehow escaping our therapies. And so when we do these studies, we're actually able to pinpoint those populations. Here you can see mice, and you can see the two brains on the top, the two brains on the bottom. The control has not received any chemo or radiation. The treatment has had chemo and radiation, and the same with the bottom, control and treatment. And what you can see is, if we just look at it the way that we look at patients, look at their MRIs, oh look, the tumor's shrinking. And on the bottom, in fact, you see all the red blood vessels, the tumor's losing its vascularity. So that's good. It looks like the chemo and radiation are working and shutting down the tumor. But in fact, the frightening thing that we can't see from MRIs is that as the tumors are treated, the stem cell markers are going up. So this is ominous. This means that this tumor, it's going to grow back because those stem cells are still there. And in fact, here you can see these are profiles, again, of controlled cells on the left and treated cells on the right. And you can see in this type of gene expression profiling, red is low and green is high. And what you can see is that a lot of the genes that are involved in self-renewal, chemo resistance or resistance to chemotherapy, and repairing DNA are all going up in the cells in the tumors that have been treated. So these tumors, they're mounting defense systems against the therapy, and they're surviving. They're evolving and they're surviving. And in fact, this is the most compelling part of this model system, is that you can see here in the purple on the edges of the sections of the spinal cord of the mouse here, the purple cells lining the spinal cord, those are disseminated medulloblastoma. And so you can see disseminated medulloblastoma tracking around the spinal cord. Well, the great thing is, did you see those vectors that we were building before? They have a fluorescent protein built into them called GFP, that makes the cells green. So we can actually pull those cells out. We can harvest these tissues, pull those cells out, and then we can profile them. So now we actually have a way to interrogate the cells that are spreading, that are relapsing. Those are cells we're never able to capture from patients. But this model system will allow us to see the cells that are actually capable of driving the spread of the tumor. So unless we ever start biopsying children's spinal metastases, we're never going to capture this kind of data from patients. So it's really important that we have model systems that can recapitulate what happens in human patients. So what are we going to try to do now? Now we have a model system that we can use to dynamically profile each patient's individual stem cell populations. And in one Christopher's tumor, it might have been the BMI1 positive population that was driving the relapse, but in the other Christopher's, it might have been the SOX2 positive population. Whatever population is driving relapse in the mouse model, we'll be able to report that back to the patient, and we'll be able to design a more effective targeted therapy that will work for that patient's tumor. So we're really moving towards tailoring molecular therapy to target the actual cells, and what I always say is that we want to try to pinpoint a moving target. Cancer is a moving target, and how do we capture that? And so we're hoping that this model system will provide some insights by the fact that we can interrogate at every stage of cancer evolution the stem cell populations. So there's been a lot of publications recently talking about tumoral, intratumoral heterogeneity, and the fact that tumors are made up of so many different cellular components. And I really think that applying a stem cell model system is a really good way of capturing heterogeneity, because of everything that you've learned today about how we track stem cells, about how stem cells generate hierarchies. And so if we can somehow apply that type of hierarchy to try to understand cancer, it's going to be very helpful. And in fact, the people who have made the biggest discoveries in looking at both the normal and the cancer hierarchies have all been Canadian. And so I always like to tell people, that we should be really proud of the fact that if hockey is our sport, then stem cell is our science. And you can see here all of the great Canadians that have contributed to these discoveries. And actually, I should tell you that down there on the bottom right, you can see Drs. Till and McCulloch. Dr. Till ended up being a great mentor to me, and he's still with us. Thankfully, Dr. McCulloch died 3 years ago. But that's me with them in the Princess Margaret Cancer Centre when I'd had one of my talks, one of my long coffee chats with Dr. Till about serendipity and scientific discovery. And you can also see that I'm 8 months pregnant with my second son, and that was 5 days before our Nature paper was accepted. So that was an exciting time, and it's nice to even contribute a small part of this whole landscape of Canadian scientific discovery. So with that, I'll thank you for your attention and tell you that these are all the people who actually do the work, and the reason we do this work is because of all of you. And what really keeps us all in the lab and working as hard as we work is the fact that we want to make lives better for all of my patients with brain tumors. And for that, we're grateful that we have you to support my patients. So thank you for that, and thank you for having me to speak here today.